Welcome back to the Deep Dive. Today we are wrestling with, well, one of the most persistent and frustrating clinical puzzles in cancer immunology, head and neck squamous cell carcinoma, or HNSCC. Okay, let's unpack this. HNSCC is a major global killer, affecting the mouth, throat, voice box.
Yeah.
And the big breakthrough treatment for many cancers now is immune checkpoint blockade, ICB. Things like PD-1 blockers, right? They take the brakes off the immune system, let T cells attack the tumor. But here's the problem. ICB, it's failing for about 90% of HNSCC patients.
Oh.
90%.
It's the ultimate paradox, really, because if you actually look inside these tumors under a microscope, they're often just packed, sometimes really densely packed, with the very cells we want to activate, the CD8 plus T cells, the ones that are supposed to be the anti-tumor assassin.
Right, so the troops are there, in force, even.
Exactly. So why aren't they winning the war? Why are they failing the mission when we think we've released the brakes?
And I think what we've learned really over decades of research is that the usual tools we use to answer that, well, they're just not cutting it. Things like just counting T cells or looking at a few general markers with, old school cytometry.
Fundamentally inadequate.
Yeah.
They tell you how many cells are maybe present, but they totally destroy the context. You lose track of which specific clone these T cells belong to, what's their precise functional state, and crucially, where are they? Where are they located inside that chaotic tumor environment?
Location, location, location,
That's
even in tumors.
Precisely. To break this kind of 10% response ceiling, we need much higher resolution. We need a map that connects a T cell's identity, its TCR clonality, to its, let's say, its job description, its transcriptional state, and its physical location.
Okay, so that's the mission for this deep dive.
That's the mission. We're exploring this really revolutionary multimodal profiling framework that, well, finally seems to achieve this. It combines high-dimensional flow cytometry, deep single-cell sequencing, and this cutting-edge spatially-resolved TCR profiling using the Xenium platform.
Xenium. Okay.
We're basically mapping T-cell fate at single-cell resolution within the intact tumor tissue.
All right, so section 1, taking the tumor apart first, right? Cataloging the players.
That's the starting point. They began with paired tumor and blood samples from 27 HNSCC patients. And the first step was this massive kind of chemical census using high-dimensional flow cytometry.
And we say high dimensional, we're not talking like 3 or 4 colors.
Oh, no. Think 31-color fluorescent antibody panel. So they can simultaneously identify dozens of different proteins on the surface of every single cell they analyze.
Wow. Okay, so a really detailed fingerprint?
Exactly, an immunological fingerprint of the environment. And the initial finding, immediate and crucial. The TILs, tumor-infiltrating lymphocytes, they are absolutely not the same as the T cells just floating around in the blood, the PBMCs.
So profiling the blood alone is kind of useless for this.
It completely fails to capture the unique picture inside the tumor, the dysfunction, the complexity, the tumor microenvironment, the TME. It's a whole different world in there.
And not just different from blood, but different from patient to patient too, right?
Yeah.
You mentioned heterogeneity.
Vastly different. They were actually able to cluster the patients into 3 pretty distinct groups based only on the mix of their TILs. This is that interpatient heterogeneity just laid bare.
Okay, what were these groups?
Yeah, so these 3, let's call them flow groups, are defined by the main immune cell type kicking around. First, the myeloid text high group. These patients had a lot more non-T cell stuff, specifically myeloid cells, the immune system's first responders, but they can also be really suppressive in cancer settings.
Okay. Group 1.
Second, the exhausted text high group. This is where you see just a huge proportion of those worn-out, exhausted CD8 plus T cells, and also lots of suppressive regulatory T cells, or TREGs.
The ones that actively shut things down.
Exactly. And third, the intermediate group. Kind of less pronounced features, somewhere in between the other 2.
What really struck me was the clinical link for that exhausted group. It wasn't random, was it?
Not at all. They found this pattern was much more common in patients whose tumors were P16 positive. That's often linked to HPV infection and also in smokers, and in folks whose tumors had come back, so recurrent tumors.
Why would that specific clinical profile lead to more T-cell burnout?
Well, it strongly suggests that chronic stress and prolonged antigen exposure are key drivers. P16 positivity, the HPV link, and smoking-related damage, both scenarios create these environments of just sustained inflammation and often higher mutation rates.
So the T cells are just constantly being poked.
Constantly stimulated, they're forced to divide like crazy until they just hit a wall, leading to this really profound, maybe irreversible exhaustion. It tells us the patient's history really shapes the T cell's fate before any treatment even starts.
Okay, so that's the cell populations.
Yeah.
But then they went deeper, right? Single-cell RNA sequencing, cRNA-seq.
Yep, on hundreds of thousands of T cells. This lets you look inside each cell and see which genes are turned on or off. Basically, their functional programming, their job title, if you will.
And what jobs did they find?
They identified 11 transcriptionally distinct subsets. But we can kind of simplify this into the key functional flavors. You've got the naive central memory cells, NTCM, marked by the gene CCR7. These are like the long-lived reserves, the stem-like pool.
The backups.
Right. Then the suppressive regulatory T cells, TREGs, marked by FOXP3, doing their job of dampening the response. And, critically, the exhausted cells. They express inhibitory receptors like PDCD1, that's the gene for PD1, and also ENTPD1.
ENTPD1, that's CD39, right? Tell me more about that one, because it seems important for exhaustion.
It really is. Think of CD39, the protein made by ENTPD1, as sort of a metabolic robber baron in the neighborhood. It's an enzyme on the cell surface that helps break down ATP, the cell's main energy currency, into adenosine.
And adenosine is bad news for immune responses.
Profoundly immunosuppressive, yeah. So when a T cell puts CD39 on its surface, it's not only signaling I'm exhausted, it's actively polluting the local area. It starves nearby active T cells of energy and just generally contributes to shutting everything down. It makes the TME even more hostile.
Okay, nasty stuff.
Yeah.
Now, connecting function to identity. They use paired single-cell TCR sequencing too, right? To get the unique receptor for each T cell, it's Clonality.
Exactly, SCTCR-seq. This tells you which cells are siblings, basically descendants of the same original T cell that recognize something specific.
And what did the clonal dynamics show? Who was dividing the most?
It really reinforces that exhaustion story. The exhausted clones were, by far, the most clonally expanded. Massively expanded.
The cells that fought the hardest ended up the most burnt out.
Precisely. The T cells that divided the most likely responding to tumor antigens were the ones that ended up completely exhausted. They ran the marathon and just collapsed.
And the stem-like cells.
Conversely, the NTCM cells, those CCR7 plus ones, they showed the highest diversity, lots of different clones, each present at low frequency, like a big pool of fresh reserves. And overall, yeah, T cells inside the tumor were way more clonally expanded than the ones in the matched blood samples.
Okay, but here's a classic question. Were these big expanded clones definitely tumor-specific? Or could some just be, bystanders, maybe reacting to an old flu virus that happens to be around?
That's always the challenge. How do you tell the difference? So they calculated this metric called tumor enrichment. Basically, the ratio of how frequent a specific TCR clone was in the tumor compared to the blood.
Right. High ratio means it's probably focused on the tumor.
Correct. And using this, they identified a specific bystander cluster. These were T cells whose TCRs were actually much more abundant in the circulation in the blood.
Okay, so definitely not tumor-enriched. What were they doing?
Well, transcriptionally, they expressed genes related to migration, like S1PR5, and interestingly, a gene called FGFBP2.
FGFBP2. Why does that matter?
It's been linked to tissue infiltration capacity. It potentially helps these T cells kind of chew their way through the structural bits of the tissue to get into the tumor site. So these bystanders are equipped to migrate in.
But they're TCRs. They weren't for the tumor.
Often, no. When they checked against databases, many of these bystander TCRs matched known microbial antigens, things like Epstein-Barr virus, EBV, or influenza A.
Wow. So your past infections literally shape which T cells show up in your tumor today.
Absolutely. Your immune history dictates which bystander troops are available and potentially able to infiltrate the tumor, even if they aren't there specifically for the tumor. It's a huge factor in the overall TME composition.
Fascinating. Okay, quick recap. We've used fancy flow cytometry and single-cell sequencing to catalog the players inside HNSCC tumors. We've got exhausted fighters, stem-like reserves, suppressive TREGs, and even migrating bystanders from past battles. We know their gene expression, their clonal history.
We know the who and the what.
Exactly. But that clinical outcome, that stubborn 10% response rate, it's still a mystery if we only look at bulk numbers or gene lists. We've taken the tissue apart, but now we need to put it back together, virtually anyway.
We need to understand the layout, the architecture.
Right. We've seen the who and the what. Now let's look at the where. Here's where it gets really interesting.
Okay, so everything we've talked about up to now involved, well, grinding up the tumor, right? Dissociating it into single cells, You lose all the spatial information.
destroying the very context we need.
Precisely. And this is why the use of spatial transcriptomics, specifically with the Xenium platform, is such a game changer here. Instead of making a cell soup, Xenium measures the expression levels of hundreds of genes in this study, 477 targeted transcripts at single-cell resolution, while the cells are still fixed in their original positions within the tissue slice.
So you keep the architecture intact. You see who's next to whom.
Exactly. We move from just counting soldiers in a disorganized pile to actually mapping their positions on the battlefield.
Okay, so what did this cellular geography, this map, reveal? Who hangs out with whom?
Well, they analyzed the nearest neighbor interactions across 17 different tissue sections, and there were clear patterns, sort of rules of engagement, T cells and myeloid cells. They showed a definite tendency to be closer to tumor cells compared to other immune cells or stromal cells.
They're right there engaging with the tumor.
Seems like it. Meanwhile, B cells largely kept to themselves, clustering with other B cells, forming their own little groups.
And did this positioning, this proximity, affect the T cell's behavior, their gene expression?
Massively.
Hmm.
This leads us straight to key finding 1. A T cell's transcriptional state, what it's doing, correlates really strongly with its neighbors. Its location basically dictates its job description on the spot.
How so? What was the pattern?
We saw this really clear spatial partitioning of function. T cells that were positioned right up close to tumor cells and myeloid cells. They consistently showed higher expression of activation and, crucially, exhaustion markers, genes like HAVCR2, that's TIM3, PDCD1, PD1, and the killing molecule marker GZMB, granzyme B.
So being near the tumor or myeloid cells drives activation but also exhaustion, the frontline grind.
Exactly. They're likely dealing with constant antigen exposure, maybe inhibitory signals from the tumor or myeloid cells. Now, contrast that.
What about the reserves, the stem-like cells? Where were they?
Those T cells expressing the naive memory, stem-like markers, CCR7, cell, IL7R, they were typically found enriched farther away from the tumor and myeloid cells. Often they were closer to B cells or endothelial cells or even other T cells, perhaps in more structured areas.
The environment, the neighborhood, is directly shaping their gene expression. That's huge.
It really is. It strongly suggests there might be these physical safe zones or maybe training grounds within the tumor landscape.
Safe zones, like niches. Did they try to identify these structures formally?
They did. They used an algorithm called DBSCAN, density-based spatial clustering. You can think of it like using a thermal camera to find hotspots, but here it's finding hotspots of immune cells, CD45 plus cells. It identifies these physical clumps or aggregates.
And what were these immune hotspots, these niches, made of?
They consistently found these clusters were rich in T cells and myeloid cells. Often, interestingly, with more CD4 plus T cells than CD8 plus T cells inside them. Sometimes B cells were nearby too. They looked a bit like those tertiary lymphoid structures people talk about.
Okay, and the key question. What were the T cells inside these niches doing compared to those outside?
This was really striking. The T cells expressing those crucial stem-like and memory markers, CCR7, TCF7, cello, were found predominantly inside these organized CD45 plus clusters.
So the reserves are bunkered down in the niches.
Seems like it. And conversely, the T cells expressing the activation and exhaustion markers HAVCR2, GZMB, they were mostly found outside these clusters. They were dispersed, mixed in amongst the tumor cells in the more tumor-dense regions.
Wow. So the niche acts like a sanctuary. It preserves the T cell's potential, their plasticity, while the main tumor bulk is this relentless frontline driving exhaustion.
That's exactly what the spatial data suggests.
Okay, this is a massive leap, but it still leaves one nagging question. Is it possible that, certain clones are just genetically programmed to prefer the niche, and other clones are programmed to go out and fight until they drop?
The nature versus nurture question for T cells. Does the clone's identity dictate its spatial fate, or does the spatial environment dictate the clone's fate?
Exactly. We need that final piece of the puzzle. We need to track members of the same clone and see where they end up and what they become.
And this brings us to the most technically innovative and I think conceptually groundbreaking part of this work. Single clone, single cell mapping in situ.
How do they even do that?
Remember those TCR sequences they got from the single cell sequencing earlier? For each patient, they identified the most abundant or interesting clones. Then they designed custom molecular probes like tiny specific labels that would only stick to the RNA transcripts coding for those exact TCR sequences.
Ah, so patient-specific, clone-specific probes for the Xenium platform.
Exactly. This allowed them to physically light up and track where individual members of a specific genetically identical T-cell clone were located within the tissue slice and simultaneously measure their other gene expression using the main Xenium panel.
So we're literally tracking genetic twins through the tumor battlefield. Now, immunology dogma often assumes, same clone, same function, right?
That's been a common assumption, yeah. If they see the same antigen via the same TCR, they should behave similarly.
So what did the data show when they tracked these twins? Did identity rule?
It completely overturned that assumption. This is key finding, too. Clonal plasticity is driven by spatial location. They saw, very clearly, that cells sharing the exact same T-cell receptor clone showed dramatic differences in their gene expression and were found in totally distinct spatial locations.
Whoa. So identity did not dictate fate.
Not solely, no, the environment played a massive role. If identity was the only driver, all members of a clone should have looked pretty much the same, transcriptionally and spatially, but they absolutely did not.
Can you give us a killer example from this study?
Yeah, look at patient 17. They focus on the single most abundant CD8 plus T-cell clone in that patient's tumor. What did they find? Members of this one, identical clone, were split. Some of the cells were clearly stem-like, expressing CCR7, and they were physically clustered together inside those organized immune niches we talked about.
Okay, the reserves within the clone, safe in the niche.
But then, other members of the exact same clone, same TCR, were found scattered broadly throughout the tumor-dense regions. And these cells, they were fully exhausted, high GZMB, high inhibitory receptors.
Wait, let me get this straight. The same T-cell clone produced cells acting like reserves and cells acting like burnouts, purely based on whether they were inside a niche or out fighting near tumor cells.
That is the stunning conclusion. The clone's identity was fixed, but its phenotype, its functional state, was plastic. And that plasticity was dictated by its local environment, its neighbors.
So proximity to supportive cells, maybe fibroblasts or certain myeloid cells within that niche structure, that preserved the stem-like potential.
That's what it looks like. But when that very same clone encountered tumor cells directly, probably got tons of antigen stimulation plus inhibitory signals, they mentioned things like the PVR-TIGIT interaction pathway. That environment drove it hard towards terminal exhaustion.
The environment is the puppet master, not just the clone's genetics.
It appears to be a critical decision maker, yes. Clonal identity matters for recognition, but the local signals seem to dictate the differentiation path and the spatial behavior.
And did they see this play out with those bystander clones too, the ones that weren't even specific for the tumor?
They did, which really reinforces how generally suppressive the tumor microenvironment can be. They tracked a blood-enriched bystander clone in patient 9, one whose TCR wasn't likely recognizing the tumor.
An innocent bystander, literally.
Pretty much. But even this clone, when its members dispersed out into the tumor-dense areas and got close to tumor cells, they started to upregulate some of those exhaustion-associated genes. They started acquiring signs of dysfunction.
So the toxic environment drags down anyone caught in it, regardless of their original mission.
It seems that way. The local suppressive signals in the TME don't really care what your TCR is specific for. If you hang around tumor cells long enough, you're likely to get functionally impaired.
This whole multimodal approach, combining the deep sequencing with the spatial mapping, it really drives home that just counting T cells or even looking at their bulk gene expression is largely meaningless without knowing the spatial context.
Absolutely. It highlights the limitations of previous approaches.
Okay, let's try and synthesize this. What's the ultimate clinical takeaway from this spatial revelation? How does it connect back to that 10% ICB response rate?
Well, think about that clinical variability we see. We mentioned earlier those patients who all got lumped into the same bad category by flow cytometry, the exhausted text group.
Right. Bulk analysis said they all look similarly poor.
Exactly. But now look at them spatially. Take patient 9, patient 12, and patient 13 from the study. By flow, they were similar, but spatially, night and day different. Patient 12 had these really dense, large, organized immune niches packed with T cells. Patient 9, on the other hand, had T cells that were much more dispersed, just kind of mixed in everywhere with the tumor cells. And patient 13 showed immune exclusion. The T cells were mostly kept out of the main tumor areas altogether.
So 3 patients, same bulk exhausted label, but totally different spatial architectures.
Totally different. And this architecture, this physical organization that flow cytometry completely misses, seems critical. Patient 12, the one with the big organized niches, they actually retain more T cells with that plastic stem-like, TEM-like phenotype within those niches. They had a potential reservoir that ICB could theoretically act upon.
Whereas patient 9, with the dispersed layout.
Far fewer stem-like cells were left, mostly exhausted cells scattered everywhere, perhaps much less potential for revitalization by ICB.
So the presence, and maybe the quality or organization, of these immune niches, the sanctuaries housing the plastic T cells, might be the real predictor of ICB success.
It certainly looks like a much stronger candidate than just bulk T cell counts or exhaustion markers alone. You could have tons of T cells, but if they're all like patient 9's dispersed and exhausted, the chance of a durable response might be minimal.
Incredible. It really shifts the focus. But okay, we should be fair. This is cutting edge stuff. What were the limitations acknowledged in the research?
Yeah, absolutely critical to mention. The xenium panel they used, while powerful, was still limited to 477 genes. That's a lot, but it's not the whole transcriptome, so you might miss some nuances in cell states or interactions.
Right. Not enough to do those really complex computational analyses of all possible cell communication pathways, like using cell chat fully.
Exactly. And mapping TCR spatially is still technically challenging. This work is a fantastic proof of principle and provides huge insights, but it's the first really detailed map, maybe not the final, complete atlas of every single interaction.
Understood. Still, the core principle seems crystal clear now. T-cell fate isn't just genetics, it's postcode, location, location, location.
Governed by the neighborhood, yes.
Okay, final thoughts then. So what does this all mean for the future? If T-cell plasticity, the stem-like potential we need for effective immunotherapy, is physically locked away in these immune niches, how do we leverage that?
That's the million-dollar question, isn't it?
Could we design smarter therapies? Maybe something spatially targeted? Could you, use specific signals to try and lure exhausted T cells back into those rejuvenating niches, get them off the front lines and into the repair shop?
That's one intriguing possibility, enhancing migration cues towards the niches.
Or maybe the opposite. What if we tried to sort of chemically enhance the supportive signals within the tumor-dense areas themselves? Try to build new sanctuaries or make the existing battleground less hostile so T cells can rest and recover even when they're near tumor cells.
Creating pop-up niches, perhaps? Modulating the local suppressive factors directly.
There are so many possibilities this opens up. Which approach do you, listening out there, think holds the most promise? Is it about moving the cells or changing the local environment where they already are? How do we best support those T cells stuck on the dangerous front line? Something to definitely mull over.